Okay, give me a second. That's our chemistry later. Right, we're going to start chapter 11 today, and that is on gene expression. Now, gene expression is another term for protein synthesis, which is also what we just finished learning in chapter 10 about transcription and translation. So we're going to start kind of reviewing that. Cells use information in genes to build hundreds of different proteins, each with a unique function, but not all proteins are required by the cell at any one time. By regulating gene expression, cells are able to control when each protein is, and that probably is, synthesized or made or produced, whatever. So I mentioned this in the last chapter when I talked about the purpose of protein synthesis. Like everything in your body, everything in cells, in every living thing is built using proteins, and a lot of our processes are regulated using proteins and enzymes and all of those things. They're all made the same way through transcription and translation. But of course, we don't need all of our proteins all of the time, and it would be a huge, huge waste of energy to make all the proteins all the time. And it could possibly even be damaging because you don't want, for example, certain enzymes being present when you don't need them. So gene expression and the control of gene expression is basically controlling when proteins are made.
How can cells with the exact same DNA in their nuclei perform different functions? For example, what makes a muscle cell different from a skin or epithelial cell if they all have the same DNA? Well, this is because cells express different genes and therefore different proteins. Not every gene on the chromosomal DNA is turned on or expressed. The cell expresses genes based on a number of different cellular requirements. This is called control of gene expression. When a gene is turned on, protein synthesis begins. A gene expresses its intended protein through transcription and translation. When a gene is turned off, protein is not made. Therefore, two cells that have the same set of genes can express very different proteins based on the function of that cell and which genes need to be turned on. For example, muscle cells turn on genes for actin and myosin production, whereas skin cells turn on genes for melanin production.
Okay, so that's also a really good point that you have hundreds of different types of cells in your body, and different cells have different functions. And so because they have different functions, they're going to be making different types of proteins, of course. So he gave at the end of that an example where you have muscle cells. The actions of muscles contracting is actually controlled by two proteins called actin and myosin. You'll learn more about that next year. And so muscle cells obviously will have the genes that produce actin and myosin, quote, turned on. That's what we use to say that a gene is expressed. When a gene is expressed, a protein is made. Okay, so that's kind of the process. I don't know, like, yeah. So when a gene is expressed, transcription and translation occur and a protein is made. Compared to a skin cell doesn't need actin and myosin because skin doesn't contract. It's there to provide a barrier between the inside and the outside of your body, but it does make a protein called melanin, which controls skin color. It also produces a lot of the protein keratin, which is what makes the skin cells a protective layer. So gene expression is the activation of a gene that results in the formation of a protein. A gene is expressed when it is turned on or transcribed. Now, I mentioned before what a genome is. The genome is your complete genetic material, and we need gene expression so we can control which parts of the genome are expressed and when they're expressed. So again, gene expression occurs in two steps, which are the two steps that we looked at in the last chapter. That is transcription, making RNA from DNA, in translation, making protein from RNA. Sorry, I'm just getting a message from the line group. What a mess. Okay, so gene expression starts when RNA polymerase transcribes DNA into a specific sequence of mRNA. mRNA then goes to the cytosol and is translated into protein. Okay, this is what we learned in the last chapter. What we're going to look at today is how, I don't know why my text is getting cut off, but we're going to look at gene expression in prokaryotes first. In a prokaryote, and don't forget, a prokaryote is a type of cell that does not have a nucleus and it does not have membrane-bound organelles. Oh, hold on. Oof. Okay. I was just talking about the online exams and different subjects not going to be tested. And apparently science is one of them. So you will not be having, at least at the moment, a bio or chem test. All right, anyway, back to the topic at hand. I'll worry about that later. Let me turn my phone upside down so I don't see messages. All right, so the way that, oh, okay, now I gotta let Tawana in, hold on. Bing. Okay. The way that prokaryotes control gene expression is through this diagram, this structure that you're looking at here. It's called an operon. This whole structure is part of the operon. There are four main parts, which I'll talk about. We have a regulator gene, a promoter, an operator, and the structural genes. The most widely studied operon is something called the Lac Operon because it makes lactose. But I'll talk about all of that as we go through it. So an operon is a series of genes that code for specific products and the regulatory elements that control these genes. So these are the genes that code for specific products. In the Lac Operon, this is a specific operon. And the reason why we use this as an example is because it's very widely studied in a very common species of bacteria called E. coli, which lives in the intestines of mammals. And since we are a mammal, it's applicable to our lives. So in the Lac Operon, there are three structural genes. So they're going to make three different proteins. And then you've got the operator, you got the promoter, and the regulator gene. These are the regulatory elements here. And these are the structural genes that code for specific products.
Because control of gene expression in eukaryotic cells is so complex, we're going to focus our discussion on control of gene expression in prokaryotes. Prokaryotes cluster related genes together and put them under the control of a single promoter so that they are transcribed into a single mRNA molecule by RNA polymerase. Operon is the term given to this cluster of genes. The promoter region is where RNA polymerase binds to begin transcription of the DNA template strand.
So I mentioned this when I talked about transcription before. There's a specific area where RNA polymerase recognizes to begin starting transcription. That's called the promoter. And then there's an area on DNA called the termination signal. And that's the signal for RNA polymerase that's the end of the gene. So the promoter and the termination signal are part of transcription.
To messenger RNA. Also notice that right in the middle of the promoter region is an operator region. The operator region can bind a repressor protein that will block RNA polymerase from passing and transcribing the genes downstream in the operon. The most common example of this operon model is the Lac Operon, found in bacteria. Lactose is a nutrient that is metabolized by bacterial cells as a source of glucose. There are three genes in the operon, and they are called LAC-Z, LAC-Y, and LAC-A. The proteins that are encoded by these genes transport and break down lactose. If lactose is not present, the repressor protein binds to the operator region, and RNA polymerase cannot pass. Therefore, the mRNA for LAC-Z, LAC-Y, and LAC-A are not transcribed. However, if lactose is present, it will bind to the repressor protein, inducing it to change its shape and fall off of the operator region. RNA polymerase is now free to pass and will transcribe these 3 genes into mRNA so that the ribosome can make the proteins for transporting and breaking down lactose. Bacterial cells are very efficient and use operon models to tightly regulate gene expression in response to their environment and nutrient requirements. By switching genes off when they are not needed, cells can prevent resources from being wasted.
Okay, so that's the overall idea of the Lac Operon, but I'm going to go over that again step by step. So again, the 4 parts of an operon, you have the regulator gene, the promoter, the operator, and structural genes. Don't know why that's, I think something happened to the text, obviously. Okay, so we're going to go through each part and look at what they do. The structural genes, oh, this is bad. I don't know why that got blown up. Weird. Anyway, we'll just work with it. So the structural genes, as I said, they code for particular polypeptides. In the Lac Operon, you've got 3 genes. So in the video, he said you've got LAC-Z, LAC-Y, LAC-A. They're going to code for 3 different proteins. Each of them are going to have a different role. Then you have the promoter. Again, the promoter is the segment of DNA that RNA polymerase binds to, marking the beginning of transcription for that particular gene. Ugh. I guess I'll just write it in then. The regulator gene over here codes for what's called the repressor protein. The repressor protein sits on top of the operator, which is the 4th structure. So you can see from the diagram that maybe a laser is better. So the structural gene codes for this red blobby thing. That's the repressor protein. Repress means to prevent from happening, or to, I think, yeah, just to prevent or to slow down or stop. And so the repressor protein sits on top of the operator and physically blocks RNA polymerase. Even if RNA polymerase binds to the promoter, it cannot move forward and transcribe those structural genes. So it's like a physical roadblock. So I'm going to talk about those when I talk about whether a gene is activated or repressed. So the operator is right after the promoter, or in the video, depending on the operon, it could be right in the middle of the promoter. But the whole idea is that the operator is where the repressor protein is going to bind to prevent RNA polymerase from transcribing the genes. That should be the Lac Operon. So this is the first operon to be studied. It's in a bacterial species called E. coli. E. coli lives in the intestines of mammals, of which we are one. And the structural genes for the Lac Operon code for enzymes that break down lactose, which is the sugar found in milk. Okay, so we're going to look at repression first. So repression means that the gene is not expressed. So this is when lactose is not present. So it makes, this is the whole point. So this is what I really want to focus on. The whole point of this chapter is understanding how organisms control when proteins are made. So if these structural genes code for enzymes that break down lactose, if you don't have lactose, why would you transcribe the genes for enzymes that break down something that you don't have? Okay, this is why, this is the key to gene expression, is understanding that You don't need lactose digesting enzymes if there is no lactose. So repression occurs when lactose is absent. The regulator gene is transcribed, okay, because it comes before everything else. As it's transcribed, it produces the repressor protein, and the repressor protein binds to the operator. Now, RNA polymerase cannot bind to the promoter, cannot transcribe these genes. This is for when lactose is not present. Okay, so again, the regulator gene is transcribed and translated. It produces the repressor protein. The repressor protein binds to the operator and physically blocks RNA polymerase from transcribing the genes to break down lactose. So when there's no lactose, you don't need enzymes to break down lactose. Activation is when the operon is turned on. So when lactose is present, that is when you want to make the enzymes that break down lactose, right? Because you want to break down that sugar. So when lactose is present, the lactose acts as something called an inducer, because inducer, I'll talk about that in a minute. The lactose binds to the repressor protein and pulls it off of the operator. And now RNA polymerase is free to transcribe the structural genes. Okay, so activation, the operon is turned on, which means you're going to transcribe and translate your structural genes. For the Lac Operon, the lactose, which is present, is going to bind to the repressor protein. It pulls it off of the operator because it makes it change shape, so it can no longer bind. And then RNA polymerase is free to transcribe the structural genes. So the structural genes are transcribed and translated, just like we studied in chapter 10. And as they're transcribed and translated, you are producing enzymes that break down this lactose. When the lactose is completely broken down, the repressor protein will be free to reattach to the operator and shut down the operon because there's no more lactose. So you don't need the enzymes anymore. So with the binding of lactose to the repressor protein, it is not able to bind to the operator. So here, it induces transcription. And induce means to make happen or to cause. That would be another one, to cause, induce.
The E. coli Lac Operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the Lac Operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the Lac Operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 Lac Operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the Lac Operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Okay. So there's the repressor protein. This is the
Prevents
controlling
gene expression.
The controlling region. You've got the operator. The operator and the promoter here. And so when, okay. Okay, those are two of the 3 enzymes that are made, by the way. So pause, there we go. So this is one of the enzymes that breaks down the lactose. This one basically just makes the cell membrane more permeable so that you can have more lactose coming into the cell. So during repression, you've got the repressor there, so RNA polymerase can't bind to it. But during activation, the lactose you see can bind to the repressor, which removes it. And that means RNA polymerase comes down, attaches to the promoter, produces the polypeptide chains. There's the mRNA. The mRNA goes to the cytoplasm. Actually, it's all in the same. Oh, I forgot to mention that. Because we are in a prokaryote, we don't have a nucleus. So transcription and translation occur in the same area in the cytoplasm. So all of this is in the cytoplasm anyway. So translation happens right after transcription. So here we have transcription, and then immediately here come your hamburger ribosomes, big unit and small unit, to clamp down on it and starts making the protein. So there's beta-galactosidase that goes off to digest, and there's permease, which goes, you can see, to the cell membrane to bring in more lactose so that you have an increase in the breaking down of the lactose. There's beta-galactosidase breaking down the lactose. When there's no more lactose, then the repressor is free to sit back on the controlling region. So you shut down the activation. If that makes sense, hopefully. Okay, hold on.